in comparison to the nervous system that you've been studying, the endocrine system takes a little while longer to respond. Instead of the immediate firing of a nerve to its end, the endocrine system, the organ has to release a chemical. It has to get to the bloodstream, and then travel through the bloodstream to wherever it's going. So that's going to take a while longer to start. Also, the response is going to last longer because as long as it's circulating in the blood, it will continue to stimulate anything that it has a receptor for it. In addition, it tends to be a little less targeted in that a nerve will go to a specific place and that's specifically what's targeted. In the endocrine system, any cell in the body that has a receptor for that particular hormone will be able to respond to it. You know that there are multiple ways in which cells communicate. The probably most straightforward, most rapid way is via gap junctions where one cell actually has a tunnel to another cell, so chemicals just diffuse to the next cell, and whatever signal one cell gets, the next cell gets. There's also paracrine responses, which are a little local response, or autocrine responses, where a chemical is actually working within that cell itself. Technically, a hormone is a molecule that is released from a cell, enters the bloodstream, and goes somewhere to do its job. So paracrine responses and autocrine responses aren't really technically hormones. The trick is that frequently hormones that are released from a cell enter the blood and are true by definition hormones. That same molecule may have a paracrine or an autocrine type response, so we kind of use the term hormone more generically than its strict definition. An important family of paracrine hormones is the eicosanoid family. In the eicosanoid family, membrane phospholipids, so you remember that most of the membrane is made of phospholipid molecules. There are specific membrane phospholipids that in the presence of certain enzymes can be converted to prostaglandins and to the leukotrienes. Those are families of hormones that trigger local pain and local inflammation. The reason these are important is because the whole family of nonsteroidal anti-inflammatory medications, aspirins, Advils, those all work to block the conversion to those leukotrienes and prostaglandins, stopping that local paracrine response. In the endocrine system, you need some key components. The first thing you need to have in the system is a gland that is capable of releasing the hormone. You also have the hormone, the chemical that's released into the bloodstream, and then you have a target cell. Really, target cells are any cell in the body that has a receptor for that particular hormone. So you have somewhere that's releasing the hormone itself, and then the receptor on a cell that is responding to the hormone. There are some organs in the body that tend to be mostly endocrine. So most of the job of that organ is making hormones. There are a lot of endocrine glands, though, that have other functions as well. For example, the pancreas is an organ that actually it's only a small part of that organ that makes insulin and glucagon to control blood glucose levels. The great majority of the cells in that organ are actually making digestive juices and part of the digestive system. It is also the case that a lot of organs that are not primarily endocrine function can make hormones. The heart, for example, clearly has a cardiovascular function, but it makes a hormone that's important in water retention and blood pressure. Hormones chemically can be quite different, and the structure of the hormone determines a lot about how it travels and how it acts. So some of the main classes of hormones The first class are the amines. Amines are made from amino acids. So a conversion of amino acid, it's a fairly small molecular structure. Most of these are polar. They have charged groups on them. The next group are peptide hormones. Peptide hormones, well, you know, a peptide is just a small string of amino acids. So it'll be larger than an amine, but not as large as a protein. So they're strings of amino acids. Many of those amino acids may be polar, so the peptide hormones tend to have a polar structure. Proteins, on the other hand, are long strings of amino acids folded in a particular 3D structure. These are large, and they tend to be polar. The last group is significantly different in that it's the steroid hormones. These hormones are all pretty much made from cholesterol. So you can take cholesterol and in the presence of certain enzymes, actually turn them into some hormones you've heard of before, like progesterone, testosterone, and estrogen. These are all hormones that are primarily lipid type structure, so they're non-polar. Many of the peptide and protein type hormones require some significant post-translational modification. So just the process of being translated does not provide an active hormone, and further steps, further cutting or addition of phosphate groups, for example, are ways in which the hormones need to be activated before they can be active. One of the things that can affect how strong a hormone response is how much hormone is actually released from the gland. Additionally, though, even if a certain amount of hormone is released, how many receptors are present on the target organ is a really important piece. For example, if there's quite a bit of hormone present, but there aren't very many receptors, you get a pretty weak response. You can have something called upregulation, where in fact the cell puts more receptors for that hormone in its membrane so that then it's capable of a stronger response to the same amount of hormone. Similarly, if in fact you have, say, the same amount of hormone, there's lots of receptors, you have a really strong response, it's possible for the cell to downregulate the receptors, take some of those receptors away so that the response isn't as strong. Two key ways in which hormones work. The non-polar type hormones, which primarily are the steroid hormones, although the thyroidamine type hormones are non-polar as well and can work this way. Any non-polar molecules, you remember, are capable of simply diffusing across any plasma membrane, so they can get into any cell they want to get into. The receptors for those type of hormones are present either in the cytoplasm or the nucleus, and predominantly they're transcription factors, so the binding of the hormone to the receptor that activates a transcription factor that will bind on the promoter region of certain genes and allow them, the transcription of them, to be turned on. In contrast, the types of amino acids, well, the types of hormones that are either the amine type, the peptide type, the protein type, have enough polar molecules on them that they're not going to be able to diffuse across a plasma membrane. They're stuck on the outside of the cell. So in this case, the receptors are all present in the plasma membrane of the cell, and the binding of the hormone to its receptor on the outside of the cell triggers an intracellular response that will end with some action. It could be a transcription factor, but it could be any number of other actions that the hormone triggers. Hormones last as long as they continue flowing in the bloodstream unless the liver degrades them, modifies them in some way, or unless they're eliminated. Either they could be eliminated in the urine, they could be eliminated in feces, or they can even be sweated out. You can sort of measure how long a hormone lasts. It's called its half-life or its clearance rate. You can actually take blood at intervals and see when do the hormone levels peak And then how long does it take until half of that hormone is gone? On average, a hormone could last 20 to 60 minutes, in contrast to a nervous system signal where it's less than a second. Hormones tend to interact. You don't just have one hormone released in your body. So the interactions of hormones can be synergistic, where one hormone works very well with another hormone to actually do a stronger job. An example of that is glucagon, is a hormone that will raise blood sugar levels. Adrenaline or epinephrine is another hormone that will raise blood sugar levels. Together, they significantly raise the blood sugar levels over one or the other, synergistic. Another example of how hormones work is permissive, where one hormone is required before a second hormone can work. So the first hormone may actually be triggering receptors for the second hormone. So if the first hormone isn't present, there aren't any receptors, so the second hormone isn't going to really do anything. And there are also antagonistic relationships in hormones, where one hormone does exactly the opposite of another hormone. So while glucagon might raise blood sugar levels, insulin will lower blood sugar levels, and they're doing exactly the opposite thing. When you look at control of how much hormone is released or present, most of that control is what's called a negative feedback system. In a negative feedback system, the first gland is releasing a hormone. That hormone release goes into the bloodstream and goes to a second gland. That gland responds to the first hormone by releasing another hormone or another chemical. That hormone or chemical, when it gets in the bloodstream, will actually feed back to the first gland and say, hey, there's enough. Don't make any more, stop. That's negative feedback. Here's an example of the negative feedback that's really common in a lot of the hormones that you'll be studying. The hypothalamus in the brain has a bunch of releasing hormones. Those releasing hormones trigger the anterior pituitary to release hormones of their own. Well, when the anterior pituitary releases hormones, they can feed back and turn off secretion of the releasing hormones. But not only that, the hormones that are released by the anterior pituitary will go to some end organ, say the testes, and trigger the testes to release a hormone like testosterone. That hormone from the end organ can feed back not only to stop production of the anterior pituitary hormone, but it can feed back and stop production of the hypothalamus hormone as well. So the negative feedback can actually be at several different levels to control making sure that levels of a hormone are kept at kind of an even balance. If in fact there isn't that much of the hormone that the testes make or the end organ makes, then there's nothing inhibiting the releasing hormones. And in fact, that's a signal, hey, we don't have enough. There's nothing here to stop us, so we're making way more of the hormone. We must need it.